Firstly, I'd like to begin by acknowledging the traditional owners and custodians of the land on which we meet today. The land on which we meet today, the Wurundjeri people of the Kulin Nation, and I would like to pay my respect to their elders, past and present, and the elders of any other communities who may be present today. So today is Daniel Gray's Lab Head Renewal Seminar. Daniel started his career in science at Monash with Richard Boyd, where he studied the thymic epithelial cells, and he then moved to do a postdoctoral fellowship at the Joslin Diabetes Center at Harvard University with Christophe Benoit and Diane Mattis on a C.J. Martin Fellowship. While Daniel was there, he already started to develop an interest in programmed cell death, and when he visited Melbourne, he came to see me a few times, and I was really impressed because he already had plans on what he would want to do, and he said, look, I'd like to switch my C.J. Martin return leg to your lab rather than go back to Richard Boyd. I was a bit apprehensive about that. I'm not into poaching people, so I said, look, I'll want to talk to Richard. So I talked to Richard, and Richard was very happy with that. He said, look, this is really good for Daniel's career. I hope it was. And so that was fine. But what Richard didn't tell me was that he was keeping the better half of Daniel to himself, Tracy. So we kind of missed out, but we also won. So in our lab, Daniel did extremely well. He soon took on the studies on cell death, and he also became enamored in the studies on cancer biology that were done in the division. And this led him to become a lab head 5 years ago. And today he will tell us what he has done as a lab head during the last 5 years. The title of his talk is The Janus-face of Immunological Tolerance and Immunity.
Thanks for that introduction, Andreas. I appreciate it. And today I'm only going to give you a couple of stories from the work that the lab's been doing in the last 5 years. But yeah, this title, so the Janus-face of tolerance and immunity, what's that about? Well, Janus was the Roman deity of transitions and duality, so hence these two faces. And these are really important themes for the main research question in the lab, which is around the question of how do T cells decide on whether to launch an immune response or not. Now, the state of non-responsiveness is referred to as immunological tolerance, okay? And this is a really important property. It's the reason why our bodies don't attack our own organs and commensal organisms via an immune rejection type response. Now, immunological tolerance has puzzled immunologists for over 50 years since Macfarlane Burnett articulated the clonal selection theory of tolerance, and Peter Medawar demonstrated it experimentally. But this question of how the immune system decides to attack or not has taken on a new dimension lately with the advent of cancer immunotherapy. So these immunotherapies, by and large, take advantage of tolerance mechanisms that are built into the immune system to now enlist the immune system to attack cancers. So if you look down here and sort of in terms of a balance, you could say that cancer immunotherapy tips it in favor of immunity to fight the cancer. However, it does this at the expense of tolerance mechanisms, and it's known now that immune pathologies that resemble autoimmune diseases are a major limiting factor for the use of cancer immunotherapies. On the other hand, immunosuppression, which is used to make people tolerate new tissue grafts that have come from other people, can be achieved with a variety of drugs, but they now tip the balance too far now towards immune tolerance at the expense of immunity, and immunosuppression is a major problem for people who are undergoing immunosuppression. So it begs the question then, why is immunological tolerance mechanism, is it important? Why is it necessary? And so for insight into this question, we need to go back to the genesis of T cells in the thymus here. So that's where they're produced. And the defining feature of a T cell is the T cell receptor, or TCR. And this is a receptor that's exquisitely sensitive to protein fragments that are presented by MHC molecules. And this is the primary way that T cells interact and sense what's going on in their environment. Now, the process of generating a T cell receptor in the thymus involves the recombination of the genes that encode the T cell receptor. This is a random process, and what it does is it gives rise to a massive diversity of different TCRs that can all see different protein fragments loaded in MHC. And I've represented the T cell clones that arise through this process in different colors, these little T cells inside the thymus here, but up to 10 to the 11 different TCRs can be generated through this random process of recombination. And so that means that as a population, the T cells are virtually, for sure, going to be able to identify any pathogen that might come along. And so this repertoire of T cells is exported into the peripheral lymphoid tissues, where these T cells are maintained in a quiescent state, and they patrol the body for any infection that might come along. And so when an infection does occur, what happens is this remarkably diverse repertoire of T cell receptors in the periphery will contain some clones that are capable of reacting to that particular pathogen. These cells become activated, they undergo many rounds of proliferation to make millions of copies of themselves, and these cells then move into the peripheral tissues where they can recognize and differentiate further to tailor an immune response to clear the pathogen. So this whole process is referred to as adaptive immunity, and it's an amazingly successful strategy. It's the reason why our bodies can recognize and reject pathogens that haven't even existed before. And also, it's evolved on at least 2 independent occasions, so it's a strategy that's been employed a couple of times throughout the course of evolution. However, there's a downside, and that is that the fact that this recombination of the T cell receptors is random means that it's also virtually assured that there'll be T cells arise that can recognize proteins derived from our own organs. When these cells are exported from the thymus and become activated, they too proliferate and then move into the organs, and they launch an immune response against our own tissues. And when that happens, autoimmunity ensues. So collectively, autoimmune diseases are a massive health burden, and there's over 80 different kinds of autoimmune diseases, and about 1 in 20 of us are going to get an autoimmune disease at some point in our lifetime. But the reason that most of us don't get an autoimmune disease is because co-evolving with adaptive immunity have been a range of different mechanisms that ensure immunological tolerance to our own tissues. Now, these act in 3 main sites to antagonize the activation of T cells. The first site is their production in the thymus. The second is their activation in peripheral lymphoid tissues. And thirdly, they can also intervene even after the activation steps when T cells move into the organs to prevent their function. Now, there's a catch to this, though, because these mechanisms have been co-opted by some clever bugs and cancers that enlist immunological tolerance mechanisms to evade clearance. So hence, again, these 2 themes of the duality of immunological tolerance and this concept of transitions or cooperativity between mechanisms at these different stages. And so the theme of today's talk is going to touch mostly on unpublished work that touch on the cooperation between immunological tolerance mechanisms at these various stages and the interface between immunological tolerance to maintain tolerance of our own tissues, but on the other hand, this tipping point when it tips over into a productive immunity against viruses and cancer. And so the first of 2 main stories is going to be about the dynamic control of regulatory T cell homeostasis and how that governs tolerogenic outcomes versus an immune response, in this case to viruses. And this work's been spearheaded by 2 very talented people. This is a senior postdoc in the lab, Karis Tay, and Simon Preston's a PhD student in Mark Pellegrini's lab. And they've worked on this project together for a couple of years now. So the backstory to this work came from these early studies that Andreas referred to when I was a postdoc in his lab, where we were studying processes of thymic tolerance. Now, one of the most prominent forms of thymic tolerance is the deletion, the apoptotic deletion of cells that have a TCR that might otherwise pose a risk if it came out into the periphery. What happens is these cells are induced to die in the thymus, so they never make it out into the periphery, so they're never activated and can't launch an autoimmune response. And what we discovered was that there's actually a continuum, at least 2 waves of thymic deletion that are really important for maintaining tolerance. And building on work from Philippe Bouillet in Andreas's lab, we found that there are 2 main proteins that drive this process, pro-apoptotic proteins BIM and another one called PUMA. But what was odd is when we made mice that lacked both PUMA and BIM, we found that, as expected, we saw a profusion of cells that are autoreactive, so these dangerous orange clones here that make it out into the periphery, and some of them get activated. But what was curious was that the mice, although they got some sort of mild autoimmune disease, didn't get a really drastic or severe phenotype, which always surprised us, because this is one of the most primary of immunological tolerance mechanisms. Now, one of the reasons for this was posited by work that Yifan Zhan did in Andrew Liu's lab, where he observed in the same mice that there was a marked expansion of a special class of T cells called regulatory T cells, or Tregs. These cells are characterized by the expression of a transcription factor called FOXP3, and this imparts a program on the regulatory T cells that allows them to suppress the activity now of autoreactive cells in the periphery. So these are green FOXP3 positive Tregs here, antagonizing the activation of autoreactive clones in the lymphoid tissues, but also they can intervene when cells are recruited into the organs as well. So they're a really potent immunosuppressive population, and this has generated substantial interest in using regulatory T cells for a range of clinical applications, such as reimposing tolerance in people with autoimmune diseases, or enabling the acceptance of organ transplants. But by and large, it's been relatively disappointing outcomes. There's been over 300 clinical trials on using regulatory T cells to reimpose tolerance. But the problem seems to be, and a good example is in the diabetes field, where people have injected large numbers of regulatory T cells into diabetic patients and found that within days, most of the Tregs have disappeared. So there's these very powerful homeostatic cell death mechanisms that wipe out most of the Tregs that have been infused into these patients. And so the challenge that we've taken on is to try and understand how regulatory T cells make life and death decisions so that we might better target these homeostatic cell death processes to enable their use in the clinic. So one example of what makes regulatory T cells a bit special in this respect is given in this really simple experiment here. So we've just taken young mice at steady state and injected them with the thymidine analog bromodeoxyuridine. And when you inject this into mice, what happens is cells that happen to be proliferating at that time take up the label and incorporate it into their DNA, and you can track BRDUs using flow cytometry to see how many cells had proliferated during that time. And we're looking in the periphery at CD4 helper cells, CD8 cytotoxic cells, and this class of regulatory T cells and their uptake of the label. And I hope you can appreciate that the Tregs have clearly taken up a lot of BRDU, implying a very high level of proliferation. So 10% have proliferated within a day after the BRDU injection, compared to their conventional counterparts, the cells that they're suppressing, which only a few percent take up the label. So because we're at steady state, that implies that at least 10% of these regulatory T cells have died as well. So there's a really high turnover of this special regulatory T cell population. And through work that we did in the past, we think that this is a really important property for regulatory T cells and the way that they regulate immune responses. So I'm from the Molecular Genetics of Cancer division, so no seminar would be complete without an overview of cell death mechanisms, but I'll keep it brief, don't you worry. But what I'll do is to divide it into three main pathways: the intrinsic pathway of apoptosis, or the BCL2 controlled pathway, the death receptor pathway, and this recently described pathway called necroptosis, which is an inflammatory form of cell death that will become relevant later on. But in work that we previously published, we discovered that the steady state homeostasis of regulatory T cells is maintained mainly by this intrinsic pathway of apoptosis. And it's all to do around the sensing of a critical trophic cytokine called IL-2, which modifies upstream members of this pathway. And there's an axis that's maintained by the pro-apoptotic molecule BIM, the pro-survival molecule MCL1, all acting on the activation of BAC and BAX, these critical gatekeepers for the entire pathway. But so I'll pose here how this dynamic process works under steady-state homeostatic conditions. And so these two things just represent the size of the populations. The regulatory T cells here, which are turning over at a high rate, antagonize their conventional counterparts to prevent their activation. And high numbers of these regulatory T cells have to be maintained by MCL1 levels. This pro-survival molecule inhibits the activation of BAC and BAX and keeps these cells alive. However, when this is perturbed, increased apoptosis of Tregs is driven by higher levels of BIM, which overcome this inhibition of BAC and BAX and lead to the death of Tregs and the shrinking of this pool. When that happens, the suppression that the Tregs had previously been applying to the conventional cells is relieved, and they become partially activated. Now, one of the first things that T cells do when they become activated is produce IL-2. It's a critical growth factor to support their activation. However, regulatory T cells can sense IL-2 very sensitively, and when they do, it upregulates the levels of this pro-survival protein MCL1, which favors their survival again. And because they're such an avidly proliferating population, it quickly reinstates the normal numbers of Tregs within a couple of days, and they reimpose this suppression of their conventional counterparts. So this axis allows for dynamic changes in the numbers and function of regulatory T cells, which is really important. If you think about immune responses that we undergo every day, we need to be able to tip the balance left and right in favor of tolerance versus immunity. And regulatory T cells seem to be critical for that. Okay, so that's nice, but the next question we asked is whether Treg homeostasis can be controlled under inflammatory conditions. Because if you think about clinical applications of Tregs, it's mostly going to be under conditions of inflammation when someone has an autoimmune disease or a transplant. And so we switched our attention to these other cell death mechanisms, and in particular, the death receptor pathway. The death receptor pathway is initiated when ligands of TNF receptors like FAS or TRAIL become ligated, and they assemble these adaptive proteins that culminate in the activation of caspase 8. It's referred to as an initiator caspase because it gets the whole process rolling. And what caspase 8 does when it becomes activated is it activates these downstream caspases 3, 6, and 7 that demolish the cell. So to tackle this question, we took a pretty similar approach to what we used previously, where we did a conditional gene ablation approach. So we used the Cre recombinase under the control of the FOXP3 promoter, so that we drive Cre expression only in regulatory T cells, and we use that to excise a floxed allele of caspase 8 that gives rise here in this Western blot to really nice specific deletion of caspase 8 only in the regulatory T cell population, not in the control mice and not in other cell populations from the caspase 8 delta FOXP3 mice is how I'll refer to them now. Okay, so what do these mice look like? Well, under steady state conditions in young mice, we're looking at here FACS plots of FOXP3 on the y-axis, so this characteristic transcription factor of Tregs, and CD25, which is the high affinity IL-2 receptor that Tregs express. And you can see that in the caspase 8 delta FOXP3 mice, there's a mild increase in the proportion of regulatory T cells, and that's plotted here of a number of mice, increased proportion, and about a 2 to 3-fold increase in the number of Treg cells that are circulating in the lymphoid system of these mice. The Tregs that accumulate in these mice are a little different to control ones in that their expression of a range of different markers, things like homing molecules like CD62L or CCR7 or PD-1, an inhibitory receptor, are slightly different and imply that these regulatory T cells are a bit more activated than normal. They're referred to as effector regulatory T cells, and they're very good at going to the tissues and intervening at that last step of an autoimmune reaction. What else can I tell you? So they're also equally good at suppressing conventional T cell responses as control mice. So this experiment, it's a conventional Treg suppression assay that we do in vitro. The components of the assay involve activation of conventional T cells. So if you look at this plot here, we're looking at the dilution of a dye called CTV that's indicative of proliferation of these cells that we've stimulated through their TCR. And then what we do is titrate in increasing numbers of regulatory T cells. So these are the ratios here. And you can see that in control mice, as we do that, the number of divisions that we're seeing, this evidence of activation of the conventional cells, decreases. And in the caspase 8 delta FOXP3 mice, when we take their Tregs to inhibit these conventional cells, it looks pretty much identical. They're equally good at suppressing T cell activation as control mice. And we found that that was also true in vivo when we used a different system. Okay, so then we conclude that caspase 8 activity is required to restrain regulatory T cell numbers under steady state conditions. And the most straightforward explanation for that is because we've removed caspase 8, and I just told you that it was required for the death receptor pathway of apoptosis, that caspase 8 is required to transduce these signals to kill a number of regulatory T cells at steady state. But then what about under inflammatory conditions? Okay, that was the main reason we made these mice. We thought that they might behave a bit differently. And so to address this question, we went to Mark Pellegrini's lab, where they'd established a really a great chronic viral infection system in mice, employing LCMV, and a particular strain of LCMV referred to as docile. It's a natural mouth pathogen and infects multiple organs, and it gives rise to a chronic overwhelming infection in these mice. They can't clear it, but they can tolerate it and they stay alive usually. And it's characterized by lots of inflammation, many cytokines, and that's what we thought would be the ideal proving ground for this question of what happens in the caspase-8 delta FOXP3 mice. There was a bit of a surprise. So what happens is they begin to die. So if here is a survival curve, and in the control mice, as I mentioned, they can normally tolerate this chronic LCMV infection, but we found that most of the caspase-8 delta FOXP3 mice that we infected died by about 140 days. The reason for this death is probably due to unrestrained inflammation. So here's some H&E stained sections taken from a variety of organs from control mice or caspase-8 delta FOXP3 mice that had been infected with LCMV, and we've taken these for analysis at the end of the experiment. And you can appreciate that even in the control mice, there's signs of inflammatory cell infiltrate in the pancreas and a little bit in the lung. But the amount and severity of infiltration that we could recover from these caspase-8 delta FOXP3 mice was much greater. So here we're seeing almost complete destruction of the exocrine pancreas in these mice, infiltration into salivary glands, some hepatitis and pneumonitis here in the lung as well. And that's just summarized here with these funky pie graphs where we're looking at the different sectors that represent a range of different organs, and they're shaded according to the amount of infiltration that we saw in those mice. And you can see that in general, the LCMV-infected caspase-8 delta FOXP3 mice had more organs, more highly infiltrated, indicating a much higher level of inflammation. And we could see evidence of this really early on. So even 8 days post-infection with LCMV, we could find evidence of liver damage with these serum enzyme levels that we can see in the caspase-8 delta FOXP3 mice compared to their wild-type controls. So we concluded that there's evidence of liver, lung, and pancreas pathology that is a likely explanation for why these mice can't tolerate LCMV infection and waste away and die. Okay, so inflammation's killing them, but why is that happening? Well, we were kind of surprised with this outcome. So what we did was analyze regulatory T cells shortly after infection. This is 8 days after LCMV infection. And we're looking at the uninfected profiles for regulatory T cells expressing FOXP3 here. That's what's being gated in uninfected wild-type mice or LCMV-infected wild-type mice. And you see normally there's a normal number of regulatory T cells between 10 and 15% in the spleen. However, in the caspase-8 delta FOXP3 mice, they start with this higher level of Tregs that I showed you earlier on, but after LCMV infection, this markedly drops. So we've gone to about a quarter of the proportion of regulatory T cells that you would normally recover from these mice. And that translates to lower numbers overall. So again, we're looking at the wild-type mice after LCMV infection, no change in Tregs. But in the caspase-8 delta FOXP3 mice, you've come from this very high level that they normally have greatly reduced after LCMV infection. This is to remind me to tell you that then these Tregs aren't hiding in other organs, because Karis did a survey of a range of different non-lymphoid organs and found that the Treg numbers were relatively similar to wild type. So we believe this loss is reflective of a cell death process. Concomitant with this loss of regulatory T cells, we see evidence of higher activation of the conventional cells, the ones they're supposed to be inhibiting. So these are two FACS profiles gated on normal CD4 positive T cells, so not the Tregs. And we're looking at two activation markers. The upregulation of CD44 and downregulation of CD62L is indicative of T cell activation, because these are homing receptors that move them into the tissues, like I was saying before. Normally, there's a fairly high proportion of cells responding to LCMV in wild-type mice, but this is greatly increased when we look at the caspase-8 delta FOXP3 mice that had been infected with LCMV. This much higher proportion, and that's reflected both in the CD4 cells here with these black bars in the caspase-8 delta FOXP3, far exceeding what their wild-type counterparts have, and the CD8 population. Probably the most telling index is if we plot the ratio of activated T cells to the regulatory T cells. This is the axis that controls immunological tolerance in many respects, because it's thriving off IL-2. And if we compare the ratio of regulatory T cells to active CD8s and CD4s, we find it's greatly reduced in the LCMV-infected caspase-8 delta FOXP3 mice. So it's reduced here and more impressively with the CD4s. This is only 8 days post-infection with LCMV. Okay, so increased T cell activation, but is this just all T cells, or is it also virus-specific ones or other specificities? We can address whether the T cells that would normally respond to LCMV infection are increased or decreased by using TCR tetramers, MHC tetramers that allow us to see those T cells with a T cell receptor that would respond to LCMV. And that's what Simon and Karis have done here, and we're plotting the proportion of CD8 T cells that bear a specificity of GP30, recognized GP33 from LCMV or NP396. And you can see in both cases, the caspase-8 delta FOXP3 mice have more evidence of LCMV specific T cells being activated. This is not just number, but also the type of T cells that are made. So here we're looking at the production of interferon gamma, a key antiviral cytokine, or both interferon gamma and TNF by these cells that are specific for LCMV. And again, I hope you can appreciate that there's much increased numbers of gamma interferon positive and double producing T cells specific for LCMV recovered from the caspase-8 delta FOXP3 mice compared to the controls. Now, this is where it gets even more interesting. So when we looked at the burden of LCMV in the mice by measuring the viral titers in the spleen, kidney, and brain, these are typical sites of LCMV infection, we found that the wild type and caspase-8 sick mice, so these are the ones that died by the end of that experiment, had relatively equivalent levels of LCMV burden. And this fits with what we saw shortly after infection. You had about the same viral burden in caspase-8 delta FOXP3 mice and wild type mice. The surprise came when we looked at those mice that were able to survive LCMV infection. So these are the ones that went all the way through that curve and didn't die like their counterparts. Here we found there were much lower titers of LCMV in the spleen, kidney, and brain. And so this implied that those cells that were able to tolerate this increase in inflammation were able to better control the virus compared to their counterparts who succumbed to this inflammatory immune pathology. So this, I think, is a really nice example of this balance. So now, because there's less Tregs, because they're dying, we've tipped the balance in favor of immunity and boosted the antiviral response. But that comes at the expense of tolerance, because there's a whole lot of other T cells now that are becoming activated and that can drive a lethal pathology that can kill some of the mice, but not all. Okay, so what's the mechanism of this? So why are these Tregs disappearing? So we turned our attention to this third pathway I mentioned, necroptosis. This is another genetically controlled form of cell death, but it's a more inflammatory form than apoptosis. Again, caspase-8 has a critical role to play in this pathway. And so what we did was look for the expression of downstream mediators of MLKL in regulatory T cells before and after infection. So here's a Western blot of caspase-8, MLKL, and a load-in control in sort-purified regulatory T cells or conventional cells here on the right. And we've taken these from either control mice or these caspase-8 delta FOXP3 mice that have either been infected with LCMV or not. And if you just look at the controls, the uninfected controls, you'll see that the levels of MLKL are really low. I was surprised. I was expecting MLKL to be expressed everywhere, because this is a really important cell death pathway. But it's not, at least in Tregs or conventional cells in uninfected mice. But when they become LCMV infected, these levels of MLKL go way up. And so now we can see really substantial MLKL expression. So we conclude that Tregs normally make very little MLKL under resting conditions, but the inflammation that attends an LCMV infection causes a marked upregulation of MLKL. So to test whether this now activates the necroptotic pathway in regulatory T cells only under inflammatory conditions, we tested whether the additional loss of MLKL could rescue the phenotypes that we saw in the caspase-8 delta FOXP3 mice. So we had 4 groups: wild-type mice, the caspase-8 delta FOXP3, caspase-8 deficient mice that are also deficient in MLKL, and another control here, just MLKL knockout mice. We infected them with LCMV docile and waited 8 days, and then we looked for these prototypical responses in the T cell compartment and whether Tregs are lost or not. And really, this is the take-home message here. These are FACS plots of regulatory T cells being gated because they're expressing FOXP3. In control mice in the spleen, we see about 8 to 10%. In the caspase-8 delta FOXP3, this drops to about 4 or 5%. But you can see in the MLKL caspase-8 double knockouts, we've got about 9% here, which is more in keeping with what you see in control mice and the MLKL mice, which don't have a phenotype either. So we can plot that again using this clinical index, this critical index of regulatory T cells ratio compared to active T cells, CD4 and CD8, and you can see that in the wild-type mice, it's at about 0.2% for CD8s. This drops to about 0.05% in the caspase-8 delta FOXP3 mice. But this is rescued if you knock out MLKL and caspase-8 together, both in the CD4 and the CD8 compartment. It looks more like what you see in wild-type mice. This translates to the LCMV specific response, which I showed you earlier, is much increased in the caspase-8 delta FOXP3 mice. It's back to control levels in the double knockout. Likewise, if we look at the activation of all CD8 T cells now by the upregulation of CD44, there's some activation here in wild-type mice. It's much increased in the caspase-8 delta FOXP3 mice, but brought back to wild-type levels in the MLKL caspase-8 double knockout. So we conclude in answer to this question, yes, MLKL deficiency does rescue the defects in caspase-8 deficient Tregs. And to summarize what this tells us altogether is that under steady-state conditions, caspase-8 plays a pro-apoptotic role because when we knocked it out, we had increased levels of regulatory T cells. However, and this again, to get back to Janus and this theme about this tipping point, this favors tolerance because there's now many more regulatory T cells. However, under inflammatory conditions, this role for caspase-8 changes. It's now required to provide a survival effect to antagonize this process of necroptosis because inflammation drives much higher levels of this critical effector of the pathway, MLKL. When we knock out caspase-8 under those conditions during inflammation, we reduce the number of Tregs and it tips this balance in favor of immunity, which is great because now we can clear the LCMV infection, but it's not great for those 60% of mice that die because of this unrestrained inflammation that leads to organ pathology. Okay, so just to summarize there, we've found these opposing roles for caspase-8 in regulatory T cell homeostasis when they're under steady-state conditions versus inflammatory conditions. And we think this has implications on how you might use regulatory T cells for clinical applications. So for the second part of the talk, I'm going to focus on a more traditional immune checkpoint type series of experiments in cancer therapy and tolerance, again to accentuate this balance. And this has been driven by two really talented PhD students. It was Antonio Pollicini in my lab and Emma Nolan in Jeff Lindemann and Jane Visveder's lab. So you probably heard about cancer immunotherapy, at least I hope so. And this is an example of its prominence. The Science named it the Breakthrough of the Year a couple of years ago. And it really is amazing. It's a new paradigm for the way we might go about treating different cancers. And the most prominent of these are called immune checkpoint blockades. And these actually borrow directly on insights that were made in immunological tolerance over 20 years ago. And in particular, they focus around two cell surface proteins expressed by T cells. The first is CTLA4, and this is a critical inhibitory molecule that puts the brakes on T cell activation in lymphoid organs. And the second is PD-1, which is expressed on T cells that have been activated and moved into the tissues. But it's a way of blocking their function in tissues to preserve immunological tolerance at the organs. Now, unfortunately, some of these mechanisms have been co-opted by cancers, such that they employ these inhibitory receptors to prevent a T cell response that might otherwise be directed against the cancer, which really is just another form of self, if you want to think about it that way. And so they've co-opted these mechanisms to evade clearance. What cancer immunotherapies do is they block these receptors. So anti-CTLA4 antibodies block this inhibition of activation, and so now you get more of these T cells being recruited into a response. Likewise, anti-PD-1 stops the inhibitory effects of PD-1 on the T cells in organs and allows them now to attack and wipe out or control the tumours. However, as I mentioned before, this comes at the expense of immunological tolerance, and it's a major limiting factor of the clinical use is these adverse immune reactions that are basically an autoimmune response to a variety of organs, typically the lung. So there's this association. There's been many clinical trials now done of immunotherapy in a variety of different cancers, and a very interesting correlation has emerged, and that's between the mutational load that the cancer cells have for these different cancers and their response to immunotherapy. So in this plot, we're looking at the number of mutations that are prevalent within the cancer population taken from all these different types of cancer. And you can see there's ones with very high levels of mutations and some with very low. Those with very high levels of mutation tend to respond to anti-CTLA4 and anti-PD-1 blockade. These are things like melanoma and lung cancer and bladder cancer. But the indications from some of these other cancer types has been less impressive, and I've picked out breast cancer here because based on this correlation, Shireen Loy from the Peter Mac and Jeff Lindeman made a hypothesis that perhaps BRCA1 mutated breast cancers might be particularly sensitive to immunotherapy. And the reason for that is because BRCA1 and 2 are tumor suppressor genes that are involved in DNA repair. And loss of function mutations in these genes drive very high mutational loads in breast cancer, in this form of breast cancer, and they drive about 10% of all breast cancers and 15% of ovarian cancers. So Shireen's lab examined this directly by taking advantage of a big collection of different breast cancer tissues, and they sequenced them and looked at the rate of mutations per case, comparing BRCA1 mutant people with those that have got cancers that are wild type for BRCA1. And what you can see here is there's an increase in the number of mutations that we can see on average in these BRCA1 mutant carriers compared to breast cancers that have wild type BRCA1 status. This is associated with an increase in immune response because these tumor-infiltrating lymphocytes, or TILs, these are immune cells that have gone into the cancer, are much increased in the BRCA1 mutant cancers compared to the BRCA1 wild-type cancers. And here's some really nice immunohistology that Shireen's lab did using a range of different determinants to really light up these immune-infiltrating cells here. So in yellow here, you're seeing one of the breast cancers, and in green and white and red, we're highlighting the T cells that are infiltrating and crowding around this cancer in the stroma. And here's another example here. So there's a lot of CD4 cells here that are right up against this, breast cancer. And likewise, here's another example. And what I want you to notice here is around the stroma, around this breast cancer, are high levels of this cyan color, which is PD-L1. It's a ligand for PD-1. And high levels of this engage the PD-1 receptor on the effector T cells that are surrounding the tumor and prevents them from launching an immune response to the cancer. So based on these preliminary data, Emma Nolan in Jeff Lindeman's lab tested whether immunotherapy might be effective in BRCA1 mutated cancers using a mouse model. So she grew tumours in these MMTV-Cre BRCA1 flox mice and transplanted them into recipient mice. And this just makes a synchronous cohort that she could then subject to a variety of different treatments. And these were either a standard of care chemotherapy using cisplatin, combined with either PD-1 or CTLA-4 blockade, or a combination of PD-1 and CTLA-4 blockade. And mice received 4 of these treatments, and then she just tracked the growth of the tumours in those mice. If we look at the survival of the mice, you can see that with cisplatin, you buy a little bit of extra time versus no treatment at all, which is consistent with chemotherapy. If you add anti-PD-1 or anti-CTLA-4 now to cisplatin, it has no effect. It doesn't change the survival of these mice or the growth of the tumours. However, if you have a combination of both anti-PD-1 and anti-CTLA-4 with the cisplatin chemotherapy, now you see an impressive increase in the survival of the mice. And this is just plotting now tumour growth now for every individual mice. It's a spaghetti plot, and it looks quite messy. But what I want you to notice is that the only group that has mice surviving beyond about 70 days are those that receive cisplatin, PD-1 and CTLA-4 blockade all together. And in fact, the time when most of these tumours broke through was after the last treatment. So it's tempting to speculate that if you kept treating, you might be able to maintain control of these breast cancers. Okay, so then Antonia came along to try and help understand the immunological mechanism that might be driving the response that you see in the combination versus the response you don't see in the single treatments. And so to do that, they took tumours that had been seeded, waited for them to grow a little bit, and then treated them with cisplatin, PD-1 and or CTLA-4, and then analysed the tumours to see what's happening to the immune cells that are infiltrating those tumours. And what they found is if they first look at the ratio between these regulatory T cells that normally suppress an immune response and the cytotoxic CD8 T cells, which are thought to be the main ones that kill the tumour cells, they observed an interesting change during the different treatments. So normally there's quite a high proportion of these regulatory T cells in the tumours compared to their CD8 positive counterparts, and it's possible that they're suppressing the response of these CD8s. However, this picture changes when you look at the cisplatin and anti-PD-1 and anti-CTLA-4 treated mice, where you see a big increase in the proportion of CD8 positive T cells that are now in the tumour at the expense of these regulatory T cells that would otherwise be suppressing the response. And this is greatly enhanced compared to the cisplatin plus anti-PD-1 or anti-CTLA-4 alone treatments. Another way of looking at what's happening in those T cells that are inside the tumour is to assay their activation. And for this, Antonio is using the expression of this inhibitory receptor PD-1 because it's upregulated on cells that become activated as well. And if you look at CD8 positive T cells in the tumour or CD4 positive T cells, you see a marked upregulation of PD-1 in the combination treatment compared to just cisplatin alone. And it's the same if you look at the individual checkpoint blockades as well. So we're seeing not only increased recruitment of cells, but we're seeing increased activation of T cells in the tumour. And those cells are more functional as well. If we assay the levels of interferon gamma and interferon gamma plus TNF expression in those T cells, we see that the combination of cisplatin, PD-1 and CTLA-4 blockade gives a marked increase in those cells expressing these cytokines. And it's most striking here if you look at the ones expressing both interferon and TNF. It's really the only group that has these polyfunctional T cells is the combination group down here. Okay, so to summarize this part, BRCA1 mutated breast cancers have a higher mutational burden and evidence of tumour infiltrating lymphocytes, which is consistent with this paradigm in the field about mutational load and the visibility of these cancers to the immune system. Cisplatin combined with CTLA-4 and PD-1 blockade was the only condition that would control the growth of these BRCA mutated cancers in this preclinical model. And this combination seems to be important for having a number of different effects on the immune system. So the blockade of CTLA-4 is promoting the increased activation of T cells and their recruitment to the cancer. Likewise, the blockade of PD-1 is required to get those cells to be more functional. So a net effect is we see decreased infiltrating Treg cells that would otherwise suppress. We see increased activation of T cells in the lymphoid tissues. I didn't show you this data, but we see more dendritic cell recruitment to the tumour, which presumably makes them more antigen processing. And finally, we see more of these cytotoxic effector T cells infiltrating the tumours, which are presumably controlling the growth of these breast cancers. So just to highlight those, we're having multiple effects on both activation and the function of T cells in the tumour site, and that's really important for controlling this BRCA1 mutated breast cancer. So this is nice justification for exploring the idea of a clinical trial that might combine this immunotherapy with cisplatin. However, there's a big question mark here about the tolerability of such a treatment, because I mentioned before there's probably an increased chance of these adverse immune reactions, these autoimmune responses, because now we're tipping the balance too far away from immunological tolerance to favour this anti-cancer response. So in the last little bit of time, I'm going to highlight another synergy that Antonio discovered in the lab between thymic and peripheral tolerance now, highlighting both PD-1 and a really important protein in thymic tolerance, the autoimmune regulator, or AIR gene. And this is going to highlight an important synergy between these very first stages of immunological tolerance, the production of T cells in the thymus, and their final functional capacity in their target organs here to drive an autoimmune response. So I should tell you a little bit about AIR, this autoimmune regulator, and it's important in thymic tolerance because for a long time there was this problem in the field. No one understood why thymic tolerance was so good. The way it works, we have to zoom in and we look at antigen-presenting cells in the thymus present peptides to developing T cells, and if they have a TCR that was randomly generated, remember, if that interacts with too high an affinity with these self-peptide MHC complexes, they're destined to die. They get upregulate BIM and that kills them. And this is the process of thymic deletion I was referring to earlier. However, the question was, how does the thymus, which is a lymphoid organ, impose tolerance to peripheral antigens, so proteins that are only made in places like the pancreas, like the thyroid gland or the brain? So here you have a situation where there's a potentially autoreactive cell. Let's say it recognizes insulin in the pancreas, but it's never going to see that in the thymus, right? Because the thymus is a lymphoid organ. Therefore, this cell should be able to shoot right out and cause type 1 diabetes because it's not being stopped in the thymus. It turns out the thymus had thought of that because there's a mechanism imposed by this autoimmune regulator, AIR, that allows for the expression of thousands of these peripheral proteins. Things like insulin, thyroglobulin, gastric hormones, you name it. They're represented here by this special class of epithelial cell that expresses the autoimmune regulator. And this is a transcriptional regulator, not a transcription factor, but it regulates processes of transcription in a way that's not completely understood, but it de-represses all these genes that would normally be quiescent in epithelial cells to promote their expression and their presentation now to thymocytes as they develop in the thymus. So now you have a situation where insulin is being presented to this cell and it can be killed by a slightly different program that involves both BIM and PUMA, and that prevents it from coming out and causing type 1 diabetes. Here's just a pie chart of these thousands of transcripts that have been discovered being expressed by these epithelial cells that express air, and a breakdown of the different organs that they represent. And you can see that just about every organ and also developmental states are being represented by expression of antigens within these epithelial cells. So this is a very thorough mechanism for removing autoreactive T cells. So many years ago, when Antonio joined the lab, actually, we asked the question of, well, air-imposed tolerance is important, but we know it's not the whole story because an air knockout mouse doesn't always drop dead straight away. So we wanted to know whether there are peripheral mechanisms that were required to safeguard tolerance in air knockout mice. So you don't need to focus on the details here, but we took a lot of different compound mutants to try and understand whether we could see, to try and see whether we could discover an exacerbation of the autoimmune phenotypes. And to cut a long story short, and a lot of mice, we established that the combination of PD-1 inhibition or deficiency with air deficiency was a really potent accelerator of autoimmunity, to the point where most of these mice didn't make it past weaning because of an inflammatory disorder that I'll tell you about now. So here's a PD-1 air double knockout compared to a litamate control. You can see it's runted and it's wasting away, and we have to euthanize those mice. Now, the reason for this is probably, we think, an autoimmune reaction in the pancreas. So here's some H&E stained sections of pancreas, lung, and salivary gland taken from aged wild-type mice, PD-1 BIM knockout mice, which don't get sick, or PD-1 air mice, which do get sick. And you can appreciate that the pancreas is a totally different picture. You've got a lot of these lymphocytes that have infiltrated the organ, and they're destroying these pink, purple-stained acinar cells. So these are the ones that are responsible for making digestive enzymes, so forth. They're being almost completely wiped out in these PD-1 air double deficient mice. We also see increased evidence of inflammation in the lung and a little bit in the salivary gland, but appreciate these are very young mice. So we suspected if we were able to support their survival for even longer, we'd see progressively worse disease in a range of different organs. Okay, so then we decided it's important to understand the cellular mechanism for this cooperation between PD-1 and air. And so what Antonia did was she purified T cells from either a PD-1 knockout, these PD-1 BIM knockouts which don't get sick, or the PD-1 air mice to see whether they could transfer disease if we put them into an immunodeficient mouse, a RAG mouse that doesn't have its own T cells. And so the controls didn't get sick, but the PD-1 air knockout T cells were sufficient to transfer disease because these mice now began to waste away and die from this exocrine pancreatitis. If she drilled down into the subsets that were driving disease, we had a little bit of a surprise. If she purified CD4 or CD8 positive T cells from the PD-1 air mice, she found that CD8 T cells were not able to transfer disease. It was the CD4 subset of T cells, though, that were driving this wasting disorder in this transfer model. Now, CD4s is an interesting population. It's a bit of a mixed bag. It includes these regulatory T cells that I mentioned before, are important for imposing tolerance and seeing disease, but then also these T helper cells, the conventional CD4s that are important for driving immune responses. And so it became important to understand whether the lack of PD-1 in the air knockout mice was an action we were having on regulatory T cells, inhibiting their function, or whether it was required to maintain tolerance in the conventional cells and stop them from making an autoimmune response. So to address that question, we turned to a co-transfer model where we made bone marrow transplants and if we did a wild type into wild type mice, there's no disease like you'd expect. Wild type bone marrow into an air knockout gives a delayed and sort of mildly penetrant disease, which is what we expect because it's an accelerator of the air knockout disease. Likewise, if we take PD-1 bone marrow into a wild type mice, slow disease. But here's our positive control. If we take PD-1 deficient bone marrow and put that into an air knockout, we get swift disease in all mice. So then, this is where it gets interesting. What if we make a mixture now of bone marrow from wild-type mice and PD-1 knockout mice? There we have a situation where there's wild-type Tregs present, but there's PD-1 deficient CD4 conventional cells as well. And so in this experiment, we're asking, can the wild-type Tregs suppress the disease that would otherwise be caused by the CD4 PD-1 deficient cells? And if you look at this pink dotted line, the answer is no, they can't, right? Because we've got a 50-50 wild-type and PD-1 knockout situation in the AIR knockout recipients. And here we see a disease that's just as swift as what we saw in the 100% PD-1 knockout chimeras that went into AIR knockouts. So from that, we conclude that PD-1 in this setting is required to restrain autoreactive T cells, probably at the site, at the pancreas. These are T cells, though, that have escaped thymic selection processes because they're AIR deficient mice. And what it does is highlights a qualitative aspect of this tolerance, such that AIR-mediated thymic tolerance of these organ-specific proteins is essential to prevent lethal T-cell-mediated autoimmunity when we block PD-1 function. So here, what we're talking about now is a cooperation between thymic tolerance mechanisms and peripheral tolerance mechanisms that, when absent, tip the balance in favour of immunity now. And so I'd posit that this has implications on cancer immunotherapy and thinking about how organ-specific antigens that might be present in the tissues are being perceived and handled by the immune system. Okay, so then I'm just going to wrap up by mentioning, again, coming back to this slide where we're focusing on the different sites of tolerance activity. And I've highlighted some stories about AIR and thymic deletion, regulatory T cells that act at activation versus function, and how they're all really important for this balancing act that immunological tolerance mechanisms have to walk, because there are times when we need to tip the balance in favour of immunity. So I only had the opportunity to present a couple of stories from the lab, Antonia and Karis's work, but the team's made up by many more than that. So here's Julie, a postdoc in the lab, and Rima Jain and Melanie Heinlein. They're all interested in processes to do with how T cells are, how these quality control mechanisms occur in the thymus. This is Antonia. She works on the PD-1 stuff I mentioned before, but also other quality control mechanisms, CARD11 and other things in T cells and cancer. Alyssa Robbins recently joined the lab, and she's working on the importance of caspase-8 in thymic checkpoints, but also peripheral checkpoints. Claire Whedon's joined us from Mariliesa's lab in a collaboration to try and understand immune responses to lung cancer using mass cytometry or CyTOF technology. And actually, that's a technology that Karis Tay in the lab has really pioneered. And I encourage you all to come to her postgraduate seminar she's giving on Monday about CyTOF and some of its applications in cancer biology and immunology. And she's been recently joined by Tim Tra. Now, he's brand new. He only came on Monday. That's what the intranet will show you for Tim if you go and look it up. He'll be supporting Karis while she goes on maternity leave to keep some of her really important work going. But you'll just have to take my word that Tim's in the lab and he's addressing this clear gender imbalance that we have in the lab right now. So we've got a great supporting cast, though, and I have to acknowledge Andreas Strasser and Jerry Adams for their excellent support for the lab in the MGC division, and also Phil Hodgkin, another mentor from the immunology division. We've had great help from other people of the MGC division who are very generous with mice, advice, and reagents. This collaboration with Mark Pellegrini and Simon Preston I've already mentioned. We have other work that I didn't present in collaboration with John Silk and Axel. The immunotherapy work from Jeff Lindeman's lab, that was a really great collaboration. A number of others. We have a great support team here. We have people helping us with genotyping the massive numbers of mice that we generate. They're generated by these animal technicians, really talented in the bioservices facility, a lot of sorting, a lot of histology, and some great national and international collaborators, and of course the funders that put their confidence in us. And with that, I'd like to take any questions you might have. Thanks.
Okay, questions for Daniel? First.
Very cool. With the PD-1 story in thymic tolerance, do you think that it's occurring in do you think it's occurring in thymic tolerance, or do you think it's in peripheral tolerance that PD-1 is most important?
We don't know for sure. I think it's unlikely to be thymic tolerance. There are a few early papers that invoked a function for PD-1 in selection events, and they're under sort of artificial transgenic systems, and they haven't really been supported by subsequent literature. We haven't done the formal experiments, but I'd say the balance of probability is on these peripheral mechanisms that are much more well established.
Anna.
Did have one. Was there a question, Anna? Right.
John.
With the mice that survived the LCMV infection, you pointed out that the possibility that you change cell wall tolerance. Those mice, have you kept them for a long time? Have they lived perfectly normal lives afterwards, or do you just get rid of them?
No, we kill them and analyze them. Yeah, we could.
Survivor was a
survivor to
relative term.
150 days is how long they survived.
Yeah.
So it was a decent period after LCMV infection. So look, there might be other pathologies if we left them for a couple of years, but my budget doesn't stretch that far.
And the increase in mutational load in the bracket tumours didn't really seem that much. It looked like about twofold.
Yep.
That slide across all cancers is on a log scale, right? So really it's usually associated with hypermutated tumours that get a response.
Yep.
So is it that this really modest change in mutational load can have a huge effect on the immune system? Or do you think it's that there's specific epitopes associated with bracket deficiency?
I think it's probably both. So that if anything, that ERA PD-1 story tells us that the type of antigens that are being perceived really does matter. Organ-specific ones matter with respect to tolerance. And I'd say that it probably extends to cancer immunotherapy as well. So I think, yeah, but I take your point. We can't, it's hard to do the experiment to try and to make them equivalent mutational loads. But I'd suggest that it's clearly sufficient because it's likely that there's some qualitative aspect that's been changed with the neoantigens that are being presented.
Yeah, do you think you could see that if you looked at the tumours that grow out? Like you've obviously gone through a population of water.
Yeah, that would be very interesting to look at. Yeah, we haven't done that, but that's a great suggestion. There's some over here.
Len, and then Ken.
Can you elaborate any more on what occurs between inflammation and caspase 8? I mean, okay, so
Yes.
inflammation promotes the reduction of caspase 8 and kicks off necroptosis. But what is it about inflammation? What's the mechanism there?
Yeah, so we're investigating that. So we're using a number of other infection models that give us graded levels of inflammation, and we're getting some really interesting data there that supports this model. Likewise, in autoimmune settings, if you use, we make these FOXP3 DTR chimeras, delete Tregs to get an autoimmune response. We find that the caspase 8 Tregs cannot antagonize that autoimmune response. And we're trying to understand the cytokines that are driving it. Short answer is interferons, but we have to work out whether there's other players, but we think it's that upregulates MLKL and now makes necroptosis an eligible pathway.
You know what happens to interleukin-2 under those conditions? Signaling
No.
those cells?
No, we haven't looked at that. We could look at phosphostat 5 and stuff, but we haven't. But what I can tell you is if you do the same experiments in FOXP3 Cree back mice, now we've crippled that intrinsic pathway, which we believe is the main sensor of IL-2 and interprets that into survival of Tregs, they look like wild-type mice. There's no problem. And they don't clear LCMV beta or anything. It all looks the same. So I don't think, we haven't tested IL-2, but I don't think it's the critical parameter in this disease.
Ken?
You nicely explained most of the things in terms of the balance between Tregs and functional T cells.
Yeah.
You mentioned that's as if it was totally non-specific.
Right.
What's the issue about the specificity of the Treg?
It matters a lot. So I mentioned those clinical trials where they injected large numbers of Tregs into people with type 1 diabetes, right? And there was a big flop. But they've since gone back and in preclinical models now found that you can increase the potency of those Tregs if you make them specific for diabetogenic antigens. Problem is now finding out what those antigens are in people, but at least there's good preclinical evidence to suggest it matters a lot. They're much more potent suppressors if you match them up to the response that you want to. I absolutely take your point. We're just looking at large numbers here, but the specificity really will matter.
Okay, right on time. Remains to thank Daniel for a really clear and